Hi everyone, we are the group 2 and today we will do a report about gram staining. Before we begin, let me share to you our learning objectives. At the end of the laboratory period, the students should be able to perform the gram staining procedure, identify the gram staining reaction, and distinguish the fundamental shapes of bacteria. So let's begin. What is gram staining? Gram staining is one of the most basic staining procedures done in the study of microorganisms. It is a differential stain that groups organisms into gram-positive and gram-negative organisms and can aid in the basic identification of organism. It is also demonstrate the basic shape of the organism being studied. All bacilli are gram-negative except Corynebacterium, Mycobacterium, aerobic spore formers Bacillus, and anaerobic bacteria spores formers Clostridium. Aerobic and anaerobic means air. All cocci are gram-positive except Neisseria, Veillonella, and Brandbamella.
Spatial components of gram-positive cell walls. Number 1 is tectoid acids, comprise major surface antigens of gram-positive organisms and can elicit antibody response. In some gram-positive organisms such as Staphylococcus aureus, tectoid acids function for the attachment of the organism to the host cell. These also provide tensile strength to gram-positive bacterial cell walls. Polysaccharides. Polysaccharides molecules include neutral sugars such as mannose, arabinose, rhamnose, and glucosamine. It also includes some acidic sugar such as glucuronic acid and monuronic acid. Diagrammic presentation of a typical gram-positive bacterial cell wall.
Components of gram-negative cell wall. The gram-negative cell wall consists of three interconnected layers of peptidoglycan, and these are the outer membrane, lipoprotein, and the periplasmic space. Outer membrane is a bilayered structure where the inner leaflet is composed of polysaccharide. It has special protein channels that allow the passage of small or slow molecular weight hydrolyphic substances such as sugars and amino acids. The lipopolysaccharide has a complex glycolipid and it is called a lipid A. Lipid A is responsible for its endotoxin activity on the outer leaflet of the outer membrane. And the inner core is the polysaccharide. It is made up of repeat units. These repeat units also called the O antigen, which is unique for every species of bacteria. The second layer is the lipoprotein. Lipoprotein function is to anchor the outer membrane to the peptidoglycan layer and stabilizes the outer membrane. It is widely believed that nearly all lipoproteins produced by gram-negative bacteria are either retained in the inner membrane or transferred to the inner leaflet of the outer membrane. Last is the periplasmic space, a fluid space between the outer membrane and the inner plasma membrane. It contains enzymes for the breakdown of large non-transportable molecules into transportable ones, and the enzymes that serve to detoxify and inactivate antibiotics.
Let's proceed to some of the comparative characteristics of gram-positive and gram-negative. In gram reaction, the gram-positive is retained crystal violet dye and stained blue or purple, while in gram-negative, it can be decolorized to accept counterstain or the safranin and stained pink or red. In the peptidoglycan layer, gram-positive has a thick multi-layered, while the gram-negative has a thin single-layered. The teichoic acid of gram-positive is present in many, while in the gram-negative, it is absent.
First materials and equipment that we will use is glass slide alcohol, lamp microscope with oil immersion, objective, inoculation loop, and reagents for gram staining.
The procedure of gram staining. Make a thin smear of the specimen. Cover the whole smear with a few drops of crystal violet for 1 minute. Gently wash off the excess stain with water until no more stain comes off. Put a few drops of grams iodine over the smear and let it stand for 1 minute. Wash off iodine with water. Flood the smear with 95% alcohol and let stand it for 15 to 30 seconds. Repeat this step until no more color comes off with the alcohol. Wash with water, counter stain with safranin for 30 seconds. Air dry and examine in the microscope using oil immersion object. Illustrated gram staining procedure. The key is crystal violet, the dot with color purple or blue. The iodine is the dot color orange. The alcohol is the colorless dot color, and the safrani is the dot color pink. In this lab activity, there is questions about the procedures. In the picture, we need to identify the gram staining reaction and morphology shape of the following bacteria. Picture the gram stain reaction is the positive and the morphology shape of this bacteria is cocci. Next picture is the gram reaction is positive. The morphology or shape is bacilli. The last picture indicated is the gram stain reaction is negative. The morphology shape is spirilla.
I will discuss the lab activity number 2 in gram staining. In lab activity number 2, indicate the function of the different reagents used in gram staining. Reagent, crystal violet. Function, primary stain. Expected result in gram positive, purple or blue. In gram negative is purple or blue. Reagent, grams iodine. Function, mordant. Expected result in gram positive is purple or blue. In expected result in gram negative is purple or blue. Reagent, 95% alcohol. Function, decolorizer. Expected result in gram positive is purple or blue. Expected result in gram negative is colorless. Reagent, safranin. Function, counter stain or secondary stain. Expected result in grams positive is purple or blue. In expected result in gram negative is red or pink.
In the tabulation number 3 is differentiate the gram-positive cell wall from gram-negative cell wall. In figure 1 is peptidoglycan. The gram-positive cell wall is thick while the gram-negative cell wall is thin. In feature complexity, the gram-positive cell wall is less complex while the gram-negative cell wall is more complex. In feature deoxide acid, the gram-positive cell wall is present while the gram-negative cell wall is absent. In feature lipopolysaccharide complexes, the gram-positive cell wall is none and the gram-negative cell wall is high. And the last feature indicated in our tabulation is endotoxin. The gram-positive cell wall is absent while the gram-negative cell wall is present.